Fox 4 News. I'm Zach Razazadeh, and today we're going to be learning about genetically modified organisms. Now, I come to you with some combinations that scientists have tried. Number 1, spider genes were put into a goat in hopes that it would produce spiderweb protein in its milk. Next, cow genes were inserted into pigskin, which turned it into cowhide for some odd reason. Jellyfish genes were put into a pig and made its nose glow in the dark. Now this one's pretty interesting. Arctic fish genes were put into fruit and vegetable trees or vines to make them freeze-proof. This one's also really cool. Strands of dog and lion DNA were put into dog eggs to make it a lion. A lion you can have as a pet. Yeah! Now, a rubber tree and a cork tree have been crossbred to create a rubber cork tree for Weber wine cork sellers. That's all for now from me. Now to Emily, Elena, and Remy.
Thank you, Zach. I'm Emily. I'm Elena.
And I'm Remy.
And we are here to talk about the percentage of GMO drinks. Our soda that we regularly have every day, or as whatever, 33%. Our other drinks are 4%. Our soda low calories are 3%. The tea is 5%. The whole milk is 9%. The reduced fat milk is 17%. The skim milk is 3%. The fruit juice, or the 100% fruit juice, is 12%. And the fruit drink regular is 13%. Now, Remy, what is a gene?
Well, I'm glad you asked, Emily. A gene is kind of like an instruction manual for our body. There are directions for building everything that makes our body function. Genes are found in DNA. Many genes are found in one strand of our DNA. All of these genes give instructions on how to make our body and body parts function correctly. If a gene is manipulated or damaged, it could lead to many different things. Genes are important to our body. Without them, we could make uncontrollable movements and possibly not even be living. Well, that's what a gene is.
Now to Elena. You might be wondering, how is genetic engineering done? Genetic engineers must force the DNA from one organism into another. Ways they do it: using viruses or bacteria to infect animals, or use plant cells with the new DNA, coating DNA onto tiny metal pellets and firing them into the cells, injecting the new DNA into fertilized eggs with a very frightening.
Salutations, and I'm Spencer, this is my Ty, and that's Liam. Now, I'm going to tell you how to make a genetically modified organism. There are 3 main components that are required: the gene you want to transfer, the organism you want to put in it, and a vector to carry the gene into the target species cell. The steps of making a GMO are relatively straightforward, but can be challenging. The gene to be transferred from the original organism. This is usually done by restricting enzymes, which are like molecular scissors, that recognizes specific sequences in the DNA and cut it at those places. Now to Liam.
Forgot to show you a picture, man.
Oh, and my picture. And that's my picture. My topic is genetic engineering precise? Current understanding of the way in which genes are regulated is extremely limited, and any changes to the DNA of an organism can have side effects that are impossible to predict. The new gene could alter chemical reactions within a cell or disturb cell functions. This could lead to instability, the creation of new toxins or allergens, or changes in nutritional value. The technology is very crude, and scientists think that if we experiment with it, we will experience very rude results. Example, when the genetically engineered salmon are compared to a control group of normal salmon, it was found that the genetic engineering had affected the activity of a number of non-targeted genes in the transgenic fish. These changes included an increased amount of the protein halvalbumin B in the genetically engineered salmon. Halvalbumin B is a protein that has been identified as a major food allergen in fish.
That's all for tonight,
folks.
folks.
And
See you next week.
And
Join us next week for the Battle of the
Battle of the
Ages.
Ages.
Bye.